Thank you so much to the committee for inviting me to present today. And thanks to all of you for being such a wonderful and engaged audience. These have just been fantastic presentations. And I have learned so much this morning and have so many thoughts about what everyone is saying that I think I've completely forgotten what I'm here to talk about. So my slides may be as much a surprise to me as they are to you, because I've moved on to new topics now in my head. So I am here today to talk about nutritional antioxidants during pregnancy and lactation. And so I have just a couple disclaimers relative to that. And the first one would be that antioxidants was a really huge category. There are hundreds and hundreds of compounds that are classified as antioxidants. There are hundreds and hundreds of biomarkers that are used to assess antioxidant status. So my first task was to really narrow those down to the things that we would be interested in nutrition. And I was also asked to focus exclusively on developing or developed and high-income countries. And that's important to note because the literature is really completely different. There have been many, many studies of nutritional antioxidants in developing and low-income countries. And sometimes people in developing countries have used that to try and inform clinical trials or something like that. But they really are very separate issues. So for that reason, I eliminated some meta-analyses and things like that that involved developing countries so that I could focus exclusively on high-income countries today. So first of all, let's talk about why we really even care about this during pregnancy. So why are antioxidants important? What is oxidative stress? So the Institute of Medicine defines it as an imbalance between the production of various reactive species and the ability of the organism's natural protective mechanisms to cope with these reactive compounds and prevent adverse events. So what does oxidative stress do? These reactive species, they can be reactive oxygen species, reactive nitrogen species, they react in the body and generate radical intermediates. Sounds so scary. Of lipids, protein, and nucleic acid. So we tend to think of them as free radicals. And basically they get a little crazy, they run around, they're out of control, and they go around and damage DNA, they damage cells. They ultimately form these chemical end products that are the biomarkers of oxidative stress. And those chemical end products have been associated with aging. They've been associated with cancer, diabetes, metabolic syndrome, macular degeneration, cataracts. Really, the list goes on and on. So, dietary antioxidants. So, we have some mechanisms to prevent the detrimental effects that we believe are caused by oxidative stress. And our diet is one of our very, very best defenses. And I think that becomes an important point when I start to talk about the status of some of the babies that we see in our population, that this is our best defense against oxidative stress. So, like I said, this is not an inclusive list at all, the dietary antioxidants. So how am I going to narrow this down to the ones in our diet? So I use the definition from the Institute of Medicine, Dietary Reference Intakes, that a dietary antioxidant is a substance in food that significantly decreases the adverse effects of reactive oxygen species, reactive nitrogen species, or both on normal physiological function in humans. And a nutrient has to meet several criteria. It has to be found in the human diet. We have to eat it for it to be dietary. The content of the substance has to be measured in foods that are commonly consumed. So this could differ from population to population. The definition of dietary antioxidants can differ based on what population you're working with. And the substance has to decrease the adverse effects of these reactive species in vivo in humans. So which substances meet this criteria in our diet? So according to the Institute of Medicine, the antioxidants that we eat are vitamin C, vitamin E, and selenium. There's another group of compounds that influence biochemical reactions involving the oxidative process but don't strictly meet the criteria. But they are going ahead and calling these antioxidant nutrients as well. And these include the carotenoids: beta-carotene, alpha-carotene, beta-cryptoxanthine, lutein, lycopene, zeaxanthin. Coming back to vitamin E, the dietary antioxidant form of vitamin E is alpha-tocopherol. There are actually 8 different forms of vitamin E, but when we are talking about the antioxidant form, we're talking about alpha-tocopherol. And the reason for that is because according to the Institute of Medicine, it is the only vitamin E substance which meets the nutritional vitamin E requirements in humans. Now, there are other categories of dietary antioxidants that are not captured here under this criteria, and the biggest one would probably be polyphenols. So between carotenoids and polyphenols, those two groups constitute most of the antioxidant function of our diet. They provide most of the antioxidant nutrients that we eat. The Institute of Medicine does not recognize polyphenols as of yet anyway, and the other point would be that in developing countries anyway, there has been really no research on pregnant women and polyphenol intake. There is one study that talks about polyphenols in the form of green tea affecting flow dynamics in patent ductus arteriosus, and that is pretty much the extent of that. So here are we've been, seeing the DRIs a lot today. So here are the DRIs for vitamin C, vitamin E, and selenium. And I think of note on this is that the Institute of Medicine, in using the outcome that they were going to set to use to set the DRI for these three nutrients, it is based on their antioxidant properties. So those three are, this is the level of intake that should promote antioxidant function in humans. Now, this gets a little bit interesting when you get to the carotenoids, because according to the Institute of Medicine, there are no recommendations made, and the current state of research is not of sufficient strength and consistency to support any recommendations regarding intake levels at this time. So as opposed to some of the talks we've heard earlier today where we can talk about what proportion of the population is deficient, who's sufficient, what do we need to do to shift this curve, we really don't know what we're talking about with carotenoids. How much carotenoids should you eat? I don't know. What should your serum concentrations be? I don't know. Is it good for you? Probably. But we really don't know what to tell you to do about that quite yet. One thing that we do know is that serum concentrations of carotenoids are a very good biomarker for carotenoid intake. So I can usually tell how much you're eating based on your serum levels relative to the rest of the population anyway. So here we have, this would be just a picture of our cohort that we're following now in Nebraska. Does what mom eat matters? So we would say yes, that as far as these serum carotenoids go, the amount that mom eats is pretty highly correlated with the serum levels of the baby. So we've studied this in carotenoids. We've also studied this with the tocopherols, vitamin E, and the various other tocopherols. And so we see here that these are pretty highly correlated. We think that what mom is eating is transferring over to the baby. We also see that with some of the other tocopherol isoforms, but not alpha tocopherol. That is pretty tightly regulated. Mom's alpha tocopherol serum level or intake is not correlated with the baby's. The baby is pretty dependent on an independent source of vitamin E immediately after birth in order to meet their alpha tocopherol needs. Now, gamma tocopherol is a different story, and I'll talk about gamma tocopherol in a little bit. Gamma tocopherol transfers very, very well from the mom to the baby. So we know that it transfers pretty well, but the baby does not end up with a lot of the carotenoids in the cohort that we follow in Nebraska. You can see here that usually the baby's level of serum carotenoid levels will be somewhere between 5 and 15% of the mom's. So the baby doesn't end up with a ton. Now, for instance, what I told you is that we don't know what a good serum level is. So, if you look at alpha carotene, mom's 54, babies are 5. How much alpha carotene does a baby need in order to have good antioxidant defenses? We don't know. Is it more than 5 in their serum? That sounds low. I don't know. I think that if you keep in mind that babies are, especially high-risk premature babies, are exposed to a lot of situations that would induce oxidative stress, this is something that we should be thinking about with these babies. Pro-oxidants are worth mentioning, I think. I guess at this point in time, they don't technically exist, but I'll just throw it out there for our consideration. Are there compounds that function the opposite of antioxidants and possibly run around, make oxidative stress work, make inflammation work. And this is entirely possible. At this time, we have not defined any nutrition compounds as pro-oxidants, but we are doing a fair amount of work, and one that we are doing some work on in my group is gamma tocopherol. So alpha tocopherol appears to be a fairly potent antioxidant nutrition. Gamma tocopherol appears to have the opposite effect. It appears to have some pro-oxidant and pro-inflammatory properties. Now, this is of interest because it transfers so well from the baby to the mom. And gamma tocopherol, actually, that's what we eat the most of. You guys know the vitamin E food sources, right? Leafy greens, nuts, and seeds. So how well does everybody in here do on that today? Everybody had their nuts and seeds today? You know, I mean, that's a hard one for people to get sometimes. Gamma tocopherol is found in basically all processed and fast food. It's in soy oil. So any fast food chain that fries everything in soy oil, any processed food that has soy oil in it is very high in gamma tocopherol. So which do you think the American population eats more of? So let me tell you a little bit about what we have learned about the antioxidant status of our cohort. So first I'm going to tell you why we care, and then I'll tell you a little bit about them. The reason that we decided to start focusing on this in my research group at the University of Nebraska was because Nebraska, and this may surprise you, does actually not very well when it comes to health disparities. We actually rate very, very badly on the March of Dimes Health Disparity Index, which means that populations, racial, socioeconomic populations that are at risk for poor maternal-child outcomes, we don't do very well. Those populations tend to have way higher adverse events in the state of Nebraska. So we wanted to see if we could figure out why that was and what can we work on from a dietary, a nutrition end in the state of Nebraska. Our population is in Omaha, Nebraska. Everybody know where Nebraska is? Really, you know, right smack in the middle, right? Take a map, fold it in half, open it, and you're, you know, right where it folded in half is where Nebraska is. And Kansas tries to claim that, but they lie. It's Nebraska. We get that. That's one of our main claims to fame. So we are a very rural state. It's an agricultural state, but we don't grow things that are good for you. We grow corn and we make it into high fructose corn syrup, or we feed it to cows or something like that. So just because it's an agricultural state doesn't necessarily mean we have a plant-based diet. And we also have a very rural population, but I'm from Omaha, and the racial and ethnic distribution of Omaha actually mimics the racial and ethnic distribution of the United States. So we feel like we are pretty representative of the United States population. So we focused on socioeconomic factors. So let me tell you a little bit about what socioeconomic factors in our cohort of about 500 women are associated with differences in antioxidant status. So the first one is food deserts. So women in Nebraska who live in a food desert have lower serum concentrations of lycopene, beta-carotene, beta-cryptoxanthine. Not surprising, just access to food being a problem. We define food desert very, very strictly, working with geographers using each person's individual address, and they are actually mapped then to the store closest to them that sells high quality food. So we feel very confident that we are defining food deserts well. Payer source in Nebraska, at least, is a very good representative of socioeconomic status. And we just classify them as public insurance or private insurance, with private insurance individuals usually being of higher socioeconomic status. 60% of our population is public insurance. 20% of our population lives in a food desert, just to kind of give you an idea of who we're talking about here. Payer source comes up a lot. Compared to those who have public insurance, people with private insurance have higher serum concentrations of lutein, beta-carotene, also alpha tocopherol, lower concentrations of gamma tocopherol. You can see here now race. So one thing that came up very interesting with race was both serum levels and intake. So I'm showing alpha tocopherol. Vitamin E sufficiency is defined by serum alpha tocopherol levels here. And you can see that the blue bar is white, the red bar is non-white. You were much more likely to be sufficient. If you were white, you were much more likely to be deficient if you were non-white. And when we did all of our modeling and everything, the odds of being vitamin E deficient are 3.5 times higher for non-whites when compared to whites. Food security. also plays a pretty big role in the vitamin E status and intakes of our moms. What I'm showing here is intakes of vitamin E, so not their serum levels, but how much of it they actually eat. So as you can see here, again, food security is, vitamin E intake is much higher in moms who are food secure compared to moms who are food insecure, and moms who either have some degree of food insecurity, either marginal or low food security, have 5.5 times the odds of having intakes that are below the RDA for vitamin E. And as you can see, it's very low. On average, that population, food insecure moms, only met about 9% of the RDA for vitamin E. Okay, so does this matter? Does it matter what their status is? Do we really care? So I would just like to now talk about some of the outcomes that have been studied with relation to nutritional antioxidant status in pregnancy. And so basically there have been about 4 or 5 outcomes in developed countries that have been studied. And usually what happens, as you well know, is there's some observational evidence and then there's some sort of clinical trial that goes on to test that observational evidence. So the first outcome I'm going to talk about is premature delivery, which is usually grouped together, at least in this body of literature, with premature rupture of membranes. And so what you can see here is that there are 3 observational studies and kind of different methodology. They looked at different antioxidant nutrients, but overall, fairly encouraging. Higher plasma levels of carotenoids, higher, and you can see in the Kramer study, the top one, higher plasma levels of the antioxidants were good. Higher plasma levels of gamma tocopherol was associated with an increased risk. So here comes this idea of a pro-oxidant again. And then some, carotenes, vitamin E popping up again. Here's vitamin C, intakes less than the 10th percentile were associated with twice the risk of preterm delivery. So some supportive observational evidence for intakes of these nutrients. So if we come to supplementation trials, then what happened when we did this? So first of all, there was one supplementation trial that supplemented with 1,000 milligrams of vitamin C, 400 of vitamin E, and this was in Turkey. This study was done in Turkey, and it showed a delayed latency period after premature rupture of membranes. So women who were supplemented after their membranes ruptured were able to stay pregnant longer and give the baby a little bit more time where he belonged. So that was one encouraging trial. Now, however, basically, when it comes to antioxidant supplementation clinical trials in developing countries, there have been 2 major studies that have been done. One was a very large NIH-funded study that enrolled 16 centers in the United States, randomized 10,000 women or so. The other one was a very similar study done in the UK. Again, very large, designed very well. And both of them were actually powered on preeclampsia. So most of these outcomes now are secondary analysis of that data. Those studies showed no impact on preterm birth. One secondary analysis of one of those studies showed that premature rupture occurring before 32 weeks, so very, very premature rupture of membranes, was less in the women that were supplemented with the vitamins. But overall, in the general populations, the supplementation had no impact on premature delivery. Of note in those studies is that at baseline, only one study reports serum concentrations. One of those studies did not measure serum concentrations, so it's really hard to tell what was the status of those women when they were enrolled. The other study reports serum concentrations at baseline, and given the mean concentrations that they report for vitamin C and vitamin E, the women in the study would have been considered replete. So these were supplementing individuals who were not deficient and possibly not even at risk for deficiency. So in this trial, now moving on to the outcome of preeclampsia. So without even looking at the studies, what I tried to do here was color code them, just to give you a feel for what the evidence looks like. So if it's colored in red, that means that the cases of preeclampsia had lower serum levels than the controls. If it's yellow, there was no difference between the 2 groups. And if it's green, the preeclampsia cases had higher serum levels of nutrition antioxidants than the controls. And as you can see by the colors, without even reading the words, the observational evidence for antioxidant nutrition and preeclampsia is a little bit all over the place. So what happened when we tried supplementation trials with preeclampsia? The same dosage, 400 milligrams vitamin C, vitamin E, no impact on rates of preeclampsia. One study enrolled women at risk, one study enrolled women with no risk. One of the studies showed there was an increase in the rate of babies that were born low birth weight, and also the babies were more likely to have a cord pH of less than 7 at birth. Now, a secondary analysis of one of these studies showed that supplementation in smokers helped preeclampsia rates, and that you would actually maybe expect to see because we know cigarette smoke is very, very damaging to antioxidant compounds. Selenium has been looked at in a developed country with regards to a biomarker of preeclampsia. And so selenium supplementation decreased biomarkers of preeclampsia in women with the lowest tertile of selenium status. And I understand there is a clinical trial going on right now to investigate this a little further. One of the other outcomes that has been investigated is respiratory outcomes during pregnancy. And what you're seeing here is a meta-analysis, and the reason I included this was because all of the studies shown up there were conducted in developed countries. Now, this is for intakes of vitamin E, not serum levels, this is intakes. But as you can see, it does look like there is possibly a protective effect from vitamin E intake on respiratory outcomes, primarily wheeze was the main outcome that was fairly consistent across these studies. Now, of these studies up above, 4 out of 5 of them also evaluated vitamin C and showed no relationship. One did show a positive relationship between vitamin C intake and wheeze. The respiratory outcomes have also been evaluated as a secondary outcome of those other 2 trials that were conducted. And what it actually showed now in a supplementation trial is it did not impact respiratory outcomes in the offspring, and it was actually associated with increased healthcare utilization. Our group has looked at growth outcomes. We were a little bit interested in this because that's what a lot of the data in developing countries is. It talks about infant growth. So we wanted to see if we could replicate some of that in a developed country. And we actually could. And one of the things that was interesting to us was in developing nations, lycopene has been fairly well studied because it's considered to be a very powerful antioxidant. As far as nutrition antioxidants go, it packs a lot of bang for its buck. And so we studied it, and we also found that moms who had higher serum lycopene had babies that had better linear or better weight and head circumference at birth. And we also looked at the cis and trans isoforms of that to kind of tease that out a little bit more. But this is independent of any vitamin A activity. Lycopene has no pro-vitamin A activity. So we are trying to tease out whether or not these antioxidant nutrients truly have independent effects on infant outcomes. We also see this with vitamin E. We wanted to look at the different isoforms at vitamin E on infant growth. We thought maybe we would see a positive impact of alpha tocopherol and then maybe a detrimental impact of gamma tocopherol, which we did not see. We saw that if mom ate a lot of any form of vitamin E, baby had better growth. That may just be reflective of other, a surrogate marker for other dietary factors. But when we looked at the baby, it flipped. So then what happened was if mom ate a lot of alpha or a lot of gamma, baby grew better. But once baby had a lot of gamma tocopherol in the cord blood, that baby did not grow as well post-hospitalization. And the supplementation trial that has looked at infant growth outcomes says that supplementation in these women did not reduce the risk of intrauterine growth restriction. Vitamin E has been sounding fairly good here. So of course it's important to throw something out that totally derails that so that none of us get too terribly excited. Because there's been a little bit of evidence in the United States that shows dietary vitamin E intakes is higher in cases of congenital heart defects. And that periconception use of vitamin E supplements, in addition to already eating a lot of vitamin E, those babies showed a 9-fold increase in congenital heart defects. Real quickly, I just want to mention diet patterns. So does this translate over to diet patterns too? So if you eat a Mediterranean diet, most of the dietary guidelines are going to lead you toward a diet that is higher in antioxidants. So is that looking like that has positive effects? And as you can see just from my highly scientific color coding scheme here, the observational evidence for diet patterns looks fairly encouraging. That the more antioxidant type of diet pattern you have, the better either respiratory or primary, it's a lot of respiratory outcomes, some growth, they're better in the offspring. Again, the yellow being the null results and green being the positive impact of a more anti-inflammatory diet pattern. So in conclusion, there is some observational evidence that shows potential associations between dietary antioxidant status and primarily premature rupture of membranes and respiratory outcomes would probably be the two that there is the strongest observational evidence for. The problem with the supplementation trials that have been conducted in populations in developed countries are that they are conducted in replete individuals, and that if you don't have a low exposure control group. Basically, you do not have a group that is deficient in the nutrient. Some of the diet pattern work may indicate that nutrients work synergistically and that food sources may be more important than individual nutrients. And from our group's own work, we definitely believe that there are socioeconomic disparities in antioxidant status in pregnant women in the United States. There's Nebraska right there.